Magandang araw, hapon, or gabi sa inyong lahat. So, welcome to Chemical Engineering 191, first semester 2020-2021. So, I'm Elmerico E. Mojica, an associate professor at Pace University here in New York City. And I would like to share to you the experience that I have with regards to the emerging technologies in removal of antibiotic residues in the environment. So the coverage of my talk, okay, can be listed as here. And in the video that you're going to watch, there will be two videos. One video is about the antibiotics in the environment. And then the second video will be about the technology that's used to remove antibiotics in the environment, focus on wastewater treatment plants. Okay, I'm sharing to you what I did when I used nanomaterials and electrical chemical methods to remove antibiotics in the environment. So, in case you don't know, antibiotics are molecules that kill or stop the growth of bacteria. It's one of the wonder drugs chemicals that discovered that benefit mankind. And for 80 years, 80 to 90 years, okay, they played a major role in the treatment of bacterial infectious diseases, prevention of diseases, and as growth promoters in the farm industry. So just to give you some sort of a history, so from 1900 to 1980, the infectious diseases mortality dropped from 797 per 100,000 persons to 36 per 100,000 persons. Okay, and the main reason for that is due to the use of antibiotics. Now, if we're going to look at the life expectancy, 1900, it's 47 years. A year after, century after, it's around 78 years. So there's really an increase in life expectancy, and this is something, antibiotic is something to do with it. Now, for the infant mortality rate, this is for less than 1 year old. In 1900, okay, 10% of the live births will die because of infectious diseases. But a century later, only 6.7 out of 1,000 live births died due to infectious diseases. And as I told you, antibiotics is one, okay, that treat if not prevent diseases. So if you're going to look at the cause of death in 1900 in the USA, almost 50%, 53% of them are caused by infectious diseases. Okay, followed by heart disease, cerebrovascular diseases, okay, and others. But a century later, infectious diseases only account for 3%. And the one that has the highest mortality rate is heart disease and cancer. Okay, and if you're going to look at this thing, so we could say from an infectious diseases, the number one cause of death has something to do with lifestyle diseases when you're talking about heart diseases, okay, and cancer, okay, because of the many chemicals that was introduced throughout the years. And they said there's a proliferation of cancer. Yeah, and this causes one of the high mortality cause of death. Okay. Now, the present status, if we're going to look at about the antibiotics, as of 2014, I have no latest data on this one. It has been said that there's a prescription of about 268.6 million antibiotics. So that means for every 1,000 person, 849 that was prescribed with antibiotics. The Food and Drug Authority or Food and Drug Agency report the sales of antibiotics in food-producing animal as around 15.6 kilogram in 2015. And if you're going to look at the state of the world's antibiotics in 2015, two-thirds, which is around 65,000 to 100,000 tons, are used in animal livestock. So to tell you frankly, there is no, we could say, fixed data on the use of antibiotics around the world, but these are just estimate, and the state of world antibiotics are projected to that the use will rise to two-thirds by to around 105,000 tons by 2013. Now, if you're going to look at the production of antibiotics in the United States, it's around 22.7 million kilogram. And in the US, 70% of these dispensed antibiotics are used or given, okay, to healthy livestock to prevent infection and or promote growth. Now, good for the European Union countries, this practice is already outlawed. So if we're going to look at the present status on the use of antibiotics, so we could say 50.46 for them are in the European, but it's nothing to do with use in the animals. Okay, it's something to do with the use in uh, human beings. America would be around 18.58%. Okay, and if we're going to look at this, Asian has almost nothing here. Okay, so we could say we are what, Western Pacific? Can we say we're Western Pacific? So in Asia, it's not really prevalently used. Okay, now the main reason why I was interested in the study of antibiotics, aside from being my PhD study, is I read this article in Proceedings of PNAS, okay, producing National Academic of Science, okay, in 2015. And it mentioned here the present status of the use of the antibiotics. So for the graph here, that's the largest 5 consumers of antimicrobials of in livestock in 2010. So as you could say, number 1 here is China, okay, followed by the US, Brazil, Germany, and India. Okay, now letter B here, largest 5 consumers of antimicrobials in livestock as projected in 2013, 2030. So that's again China. So it's always China. There's always a saying that if it's not made by God, it is made by China. Or if it's not created by God, it's most likely be made in China. Okay. And the third one here, so the largest increase in antimicrobial consumption between 2010 and 2030. Okay. Again, China is the number 1. So what piqued my interest here is the last set of data. The largest relative increase in antimicrobial consumption between 2010 and 2030. And you have there the Philippines. So you have there Indonesia, Myanmar, Nigeria, Peru, and then you have the Philippines. So I was surprised that we are included. And the thing is, there's a really prevalent use of antibiotics in agricultural farms, especially in animal farms. And the way that they do is they hide it because they sell it as a brands like vitamins. Okay, so that means they use it as a growth promoter. Now, if we're going to look at the categories of the antibiotics, we can classify them based on the mode of action that they have. Okay, so you can have their inhibit bacterial cell wall biosynthesis as the main mode of action for antibiotics that has the beta-lactam, the cephalosporin, the glycopeptides, and the ninomycins. So example that can be included here is penicillin. So typically it's used for human therapeutic and animal therapeutic and sub-therapeutic. So when you say subtherapeutic, so that means it's used for growth promotion. And then we have also a group of antibiotics that block the 30S or 50S ribosomes. In a way, it inhibit the protein synthesis. So among here is macrolides, streptogramins, chloramphenicol, fusidic acid, tetracyclines, which I have used in my study, lincosamides, aminoglycosides, and oxazolidinones. So this is also used for human therapeutic and animal therapeutic therapeutic uses. Quinolines and rifamycins are classified as one that blocks the replication of the DNA. So you use this for UTI and systemic and respiratory infections. You can also include antifungal azoles and folinies to disrupt the integrity of cell membranes here. So it treats the fungal infection and topical applications for eczema. Okay. And then the last type of antibiotic with the mode of action would be the one that antagonizes the metabolic processes. So here we include the sulfonamides, which I also studied, and trimethoprim. So you use this for humans, livestock, aquaculture. Trimethoprim metaxazole are often used for urinary tract infection. I think you can see it, but it should be for urinary tract infection. Now, if we're going to look at the classification of the antibiotics, we can classify them as either bacteriostatic or bactericidal. So when we have bacteriostatic, okay, so when we're talking about bacteriostatic antibiotics, so these inhibit or delay bactericidal growth and replication. So this would include chloramphenicol, erythromycin, clindamycin, sulfonamide, trimethoprim, and tetracycline. On the other hand, when we're talking about bactericidal antibiotics, these are drugs that target or kill the target organisms. Okay, and this would include aminoglycosides, beta-lactams, vancomycin, quinolones, rimpafim, and metronidazole. Now, some antibiotics can be both bacteriostatic and bactericidal, depending on the dosage, the duration of exposure, and the state of the invading bacteria. Okay, we can also classify antibiotics as we could say broad spectrum. When we say broad spectrum antibacterials or antibiotics, they're active against both the gram-positive and gram-negative organisms. Okay, and this includes the carbaphenems, chloramphenicol, third generation fluoroquinolones, second, third, and fourth generation of cephalosporin and tetracyclines. Now, there are also antibiotics that are of narrow spectrum. So these have limited activity and are primarily only useful against a particular species of microorganisms. Okay, so some of them we could include here the penicillin, lincosamides, glycopeptides, streptogramins, and rifamycin. Now, glycopeptides and bacitracin are only effective against gram-positive bacteria, whereas polymyxins are usually only effective against gram-negative bacteria. Okay, so if we're going to look at the fate of the antibiotics, usually here is how antibiotics end up in the environment. So we could start antibiotics either through the use in human therapy or therapeutic purposes, or how antibiotics is used in veterinary medicine. Okay, so what happened when humans use it, only a portion of it is being absorbed by the body. Almost all of it are metabolized to other products and excreted by our body. So when it is excreted, here in the US, we have this sanitation system that is connected to the wastewater treatment plant. So the excrement, the urine and the feces that come out of your body will go to the wastewater treatment plant. Now, in some of these things, the wastewater treatment effluent, it can mix with the biosolid. Okay, some of them will be discharged and go to the surface water or the groundwater. Some of them will go to the soil. Okay, now we all know that in wastewater treatment plant, they treated the water not because of the presence of the antibiotics metabolites. They treated it to treat for other purposes, for other chemical that they use. And since not all of these antibiotics are treated, it can go back, okay, in the form of the drinking water. So some of these metabolites can go back and then utilized by human. Okay. Now for some, if it goes to the surface water and the groundwater, it can go to the crops or the aquatic system or the fishes that we have. And they can be contaminated by antibiotics and we can intake them, okay, directly. Now, in terms of the veterinary medicine uses, so as I told you, they use for therapeutic and sub-therapeutic level, okay? So the livestock, like human beings, they will excrete their waste with some antibiotics metabolized in it. Now, what do we do? Okay, we usually use this manure as fertilizer. So this is just, we could say, a warning to those who are fond of these organic products. Okay, so most likely they also have antibiotics residue in them because they use the manure as a fertilizer. So they spread it. So some of the manure is used in aquaculture. If I'm not mistaken, in the tilapia farming, they use the manure to feed the tilapia fingerlings. Okay, so since it is in the environment, the soil can be contaminated, the surface water and the groundwater is also contaminated, and then it can be absorbed by the crops and the fishes, and the crops can be obtained by the livestock, which indirectly can be contaminated and be ingested by the human being. So this is the fate of the antibiotics in the environment. So if we're going to look at this at the human input, we excreted therapeutic antibiotics and the metabolites. So as I have mentioned, many excreted directly in the amount that is more than 40% of the ingested dose. And here, some of them has a program that if you're not going to use your antibiotics or you have not used it or you didn't finish the usage and you still have some tablets there, okay, some pharmaceuticals, they can take it back or you can bring it back to them. Others will just dispose this unused of antibiotics, flush it down the toilet. So it goes to the wastewater treatment. So washing topically applied antibiotics as another source. Okay. Now all of these go to the septic systems or the municipal wastewater treatment plant. Unfortunately, as I told you, it is its removal is inefficient because it's not target the antibiotic residue, and this becomes the breeding ground for antibiotic resistance organisms. For the veterinary uses, we could say there are four types of which antibiotics is used in livestock operations. The first one is something to do with therapy or therapeutic purposes, antibiotic for treatment of clinical diseases. They're also used for control. Okay, so antibiotics as administered to a herd or flock in which morbidity or mortality has exceeded the baseline norm. So they just like to control any outbreaks of diseases that they have in their animal population. And then they also use it for prevention, okay, wherein antibiotics is used in animals considered at risk, but where individuals do not show signs of disease. Then the thing where it is mostly used, at least here in the United States, is as growth promotion. So it administer antibiotics administered over a period of time, usually as a feed additive to growing animals. So the way that they use it as a growth promoter, they only use a subtherapeutic amount. So the idea on why it is used is still debatable, but they said its use has something to do with the improvement of the microorganisms in the gut of the animal that improves or make growth faster. Okay, so there's still a lot of theories on how it is effective as a growth promoter. Now, for instance, just to give you how cattle here in the US is being administered with different types of antibiotics. So penicillin is used in it, or flunixin is used as anti-inflammatory and analgesic. Antibiotic like sulfadimetoxin, so this is a sulfonamide, is also introduced on them. And then we have the gentamicin, okay, and another sulfametocin, and then tilmicocin, so this is antimicrobial used for respiratory diseases, and then you have the tetracycline and ampicillin. So if you're going to look at this one, this is for therapeutic purpose, okay? And there are news wherein it's positive news lately. If I'm not mistaken, this was 3 years ago that FDA report shows decline in the use of antibiotics in animals. Okay, so according to this report, from the Center for Infectious Disease Research and Policy, there's a drop by 10% from 2015 to 2016, and that includes 14% drop in the use of medically important antibiotics. So that's the good news. Okay, but then what we have is, despite the rise of the superbug cases, the report still finds the rampant antibiotic use in the pork industry. They're still using it. Okay, and this photo was taken from our canteen, and they always advertise their antibiotic-free chicken or antibiotic-free pork. So that means they did not use this antibiotic as growth promoter. And the fast food chains place support for antibiotic-free meat, but some promises turn up empty. Okay, so here in the US, the consumers advocate for the food that they eat. So they have a strong opinion about the food that they take inside their body. So they ask the restaurant to do it. Okay, so you have restaurant like Panera and Chipotle. Okay, they reported the use of the thing on this what we call antibiotics, and all of these chain, okay, or one way or another, they reported the use of antibiotic chicken or pork or meat that contains antibiotic. But because of this thing, okay, some of these chain of fast food, some of these fast food chains became aware that their consumers, okay, know the food that they're eating. So some of them try to start using this antibiotic-free chicken or antibiotic-free pork. Now, if you're going to look at the amount of the antibiotic that is introduced in the animal, okay, or the veterinary impacts. So the range of the amount of antibiotic residues depends on the animal species and their age. So, you know, some animals are big, some animals are small. So they said it depends on the size. So the cow or the cattle that I have shown earlier in one of the slides, they have a lot of antibiotics. So they have a lot of dosage of antibiotics that's being injected to them compared, let's say, with the chicken. Okay. So if we're going to look at here, total veterinary antibiotics present in swine, cattle, and poultry manure ranges from 1 microgram per kilogram to 1,420 milligram per kilogram. And animals release 30 to 90% of the consumed antibiotics in urine and manure, which is good if you're the meat, but it's not good when you are the environment. Okay? And the tetracycline treated animals excrete between 70 to 90% of the antibiotic administered via urine and feces. Okay? Now, if you're going to look at the half-life of the antibiotics in soil, so this is some of them as reported from this paper. So as you could see here, the tetracycline, like oxytetracycline and chlortetracycline, they have a half-life that is less than 30 days. Penicillin, okay, also has only 7 days. Now, sulfametocin, less than 20 days. Now, the one that has the longer one are, we could say here, virginomycin. And if you're going to look at the potential for bioaccumulation, some of them can be bioaccumulated by plant. Okay, and the concentration that these antibiotics were found in the soil are listed here. So from as low as 2.5 to 50 microgram. Okay, and this one is also around, if I'm not mistaken, around 17 microgram per gram, if you're going to convert the thing here. So antibiotics can always be, we could say, detected in soil, especially if the soil is near animal farm. Okay, so another thing that they look is the KD value. So the KD values, this is the constant. So this has something to do with the distribution constant. So what information you can get from here? The higher the value of the KD constant, the more, okay, that they remain persistent in the soil. So tetracycline, as you could see here, 0.32, 1030. That means some of them are mobile and some of them are persistent. And it has something to do with whatever side group that they have. They have the same functional group, but there are some side chain that makes them mobile or persistent. Sulfonamides are usually mobile. So when you say mobile, they move from one place to another. Okay, fluoroquinolones, unfortunately, is one of the most persistent antibiotics. So they will stay for a longer time. So if you're going to ask, okay, what does this mean? If you are mobile, you can move by bodies of water. But if you're persistent, you will remain in the sediments or the soil part, okay, of the material. Tylosin, it has also some portion. So if we're going to look at the KD values that we have here in one study, tetracycline is the most persistent, followed by the norfloxacin. Okay, so most persistent, less persistent, erythromycin is less persistent than fluoroquinolone, norfloxacin. Chloramphenicol is less persistent. Sulfametocin is the most mobile. So that's the information that the slides tell us. Okay, now if we're going to look at the antibiotics in soil, so the animal manure is the main source of the antibiotics. Why? Okay, because it is applied as fertilizers in the soil. So it can serve as a non-specific source of antibiotic residues in farmlands and aquatic environments. That's why I told you, those are fun of this organic stuff. Just make sure they don't have any farm manure that they have there, because most of the farm manure animals, most of the manures from farm animals that are treated with antibiotics, they're going to release antibiotic residues. The tetracyclines are found to be persistent in the soil. So in one study, 295 microgram per kilogram of tetracycline and 39 microgram per kilogram of chlorotetracycline. And then the amount of the sulfa is much less, around only 2 microgram per kilogram sulfamethoxine, because I told you this is a mobile antibiotics. You don't see them persist in soil. And then 50 microgram per kilogram tylosin detected in stem cell samples. So this is a study where you have these mixtures of antibiotics, and they try to monitor the movement of these antibiotics in the soil. So another study says leaching field study using clay soil showed the absence of oxytetracycline and tylosin and the presence of the sulfachlorpyridazine in the 20 to 30 centimeter depth soil sample, or the leachate. So what does it mean? Okay, so if you look at the movement of the antibiotics in the soil, okay, the sulfachlorpyridazine, we're going to move downward 20 to 30 centimeter depth soil. And if you're going to collect the water coming from this, you can detect the presence of the sulfa antibiotics because it's mobile. Okay, so if we're going to look at the concentration of the antibiotics from one source to the different environmental samples, so we could say the highest source of antibiotics are the manufacturing plants. It makes sense because that's where they manufacture. Okay, so one plant in India reported around 31 to 920 milligram per liter. And then its concentration decreases as it goes to the wastewater. Okay, and then some of them end up in soil. Okay, so from 12,000 microgram to kilogram, it's reduced to 200 microgram per kilogram in soil. And then some of them will be in the sediments. That's around 25 microgram per kilogram. And then, as I've told you, in aquaculture, some of fish farmer, they use manure to feed the fingerlings. Okay, so that introduces you around 500 to 4,000 microgram per liter. Surface water, okay, that's around 1,900 nanogram per liter. And then the groundwater would be around 400 nanogram per liter. Drinking water, they found out, okay, as high as 200 nanogram per liter. In China, most of the papers that they have, the levels are just so high. Okay, I think that country is just so polluted with antibiotics. Okay, now in water, I already discussed some of them from the previous slides about the concentration of them. So usually the concentration of antibiotics in water are the one coming from the hospital. Their effluents are 10 to 1,000 times higher compared to those in municipal sewage and sewage treatment plants. But unfortunately, I don't think we treat the hospital waste in our country. Some of them just goes on the bodies of water. Okay, so that's going to be problematic. Now, 90% of the antibiotic inputs in the wastewater treatment plants comes from hospital. So this is if you have the wastewater treatment plant. So the main source of antibiotics are those coming from the hospital, because this is where they're being prescribed or where they have a surgery. So when they excrete it, it will go to the wastewater treatment plant. As shown here, wastewater treatment plant are the final place where antibiotics can be treated before entering the natural system. But unfortunately, we don't have that in the Philippines, right? Do we have any wastewater treatment plant? I think what we have is the Pozo Negro or Malabanan excavation site. I don't know if that's still available at this time. That's the one that was very popular when I was there, maybe 10, 15 years ago. Okay. Now, the wastewater treatment plant can become as anthropogenic sources of antibiotics. So what do we mean by anthropogenic man-made sources? Okay. And I just like to show to you some of the reported level of antibiotics in some bodies of water. The Victoria Harbor in Hong Kong, erythromycin was found to have this concentration around 5.2 to 1730 nanogram per liter. Sulfamethoxazole is found to be around 47 nanogram per liter. The famous San Francisco Bay, where you have this San Francisco Bridge, they found 270 nanogram per liter of sulfamethoxazole. The Creek seawater, they found it to have 128 nanogram per liter amoxicillin. The Lake Geneva, they found it to have around 130 nanogram per liter sulfamethoxazole. The Maumee Bay here in Lake Erie, USA, they found it to be around 211 nanogram per liter of sulfamethoxazole. You want a much higher level of these antibiotics, you go to some papers by Chinese authors. The levels that they have, they're so high. Okay, I didn't put it here, but those are the developments that come out when I always review letters about what we call the papers coming from China. The levels that they have was just so high. I'm thinking that, is this a sediments or bodies of water? Because the level that they have is usually found in the sediments. Okay, now the groundwater, which is usually the source of our drinking water. Okay, when they did these national reconnaissance studies of the occurrence of pharmaceutical compounds in groundwater and drinking water waters, it confirms the presence of the antibiotics, and the one that is common are the sulfa. So in some of them, they found 1.11 microgram per liter of sulfamethoxazole and 0.36 microgram per liter sulfamethoxine, and lincomycin of around 0.32 microgram per liter were found present on some of the 47 groundwater sites that was sampled. And then another study, they found, okay, the sulfa 161.1 nanogram per liter in source water and 8.2 nanograms per liter in treated drinking water present in some of the 25 drinking water treatment plants from 24 states here in the US. Okay, so that's the problem that we're going to have. So COVID, we could say, is the immediate one, but this is the main problem that we have here in the US. How sure are we that our water, our drinking water is clean? And antibiotic residue is just one of them. We also have some problems with these halogenated materials. Okay, so these halogenated materials, just to give you an idea, what do you use to treat plant? Chlorine. So they found out that some chlorinated and fluorinated materials are formed. If you heat the water to drink your coffee, okay, make sure you only use it once. If you're going to reheat it again, there's already some chlorinated and fluorinated materials that you have there. And that is not, they said, safe. Okay, so that's the problem that they have here. How to fix this antibiotic residues and fluorinated or halogenated materials that are formed in drinking water. Now, if we're going to look at the ecological impacts, okay, how are the antibiotics affect this thing? Okay, so if we're going to look at the environmental contamination by antibiotics, it may have profound ecological impacts or effects at several levels, but the increased emergence and distribution of antibiotic resistance in pathogenic bacteria is by far the most important issue. So there's been an increased emergence and distribution of these antibiotics, especially the pathogenic bacteria. So that's the one that is the most important issue. And constant exposure to antibiotics provides an environment that is conducive for selective pressure towards antibiotic resistant microorganism and the antibiotic resistant genes or ARG that they carry. So even very low concentration of antibiotics and their metabolites may potentially stimulate this antibiotic resistance gene. Okay, so these are just some of the effect, the ecological effect on the presence of the antibiotics. It promoted microbial growth and activity. There's an increased concentration of the dissolved organic carbon in soil. And in some of them, they saw a shift in soil bacterial community structure. In some, they reported the stimulated soil basal respirations. And then there's also reduced population in target and non-target organism. They found that there's a negative effect on the soil biomass and microbial activity. And in several papers, the presence of these antibiotics reduced the rate of denitrification, carbon source utilization, biogas production, and other processes. Okay, so for a specific effect here of ciprofloxacin, a fluoroquinolone effect on the algae. So with triclosan and tergito NP, it affects the rate of algal biomass production and algal community. So they found out that this antibiotic together with these compounds, they cause significant increase in the growth of Synedra and a decrease in the growth of Nabicula. So these are just algae organism. Okay, in the soil where you use the manure as a fertilizer. So when you apply, okay, manure. So usually there's a nutrients that you have there, the nitrogen, phosphorus, potassium, and organic carbon. But they're not the only sources that you have there. You can also have antibiotic substances, heavy metal, disinfectant, resistant bacteria, resistant gene, and mobile genetic elements. And it's going to affect the antibiotic resistance. What happened there, the manure nutrients will stimulate the microbial activity and the horizontal gene transfers, as I told you, some of the effect that it has. So the bacteria applied with manure increase the resistant gene abundance and the overall increase of the resistant bacterial population. The effect on the soil function, the antibiotic substances can decrease the resilience of the soil microbial communities against the disturbances from the manure application. And the soil function can be restored by functional redundancy. Now, in terms of the rhizosphere here, they found out the effects of the root exudation. So what does this mean? The root exudate stimulate the microbial activity and horizontal gene transfer, which is plant specific. It increased dissipation of antibiotic substances. It also lower increase in relative abundance of resistant genes and mobile genetic elements than in bulk soil. So antibiotic selection continues due to the stimulation of microbial activity. But that's not the only problem that you have. Okay? Plant can uptake the antibiotics that you have in the soil. So whenever there's antibiotic from wastewater irrigation or organic waste application, there could be absorption, degradation, and leaching of these materials. And unfortunately, the root can take it up. Okay? And it can undergo what? Translocation, bioaccumulation, and if the human eat it, okay, you are introducing antibiotics to the human being. And they reported the presence of these antibiotics in different plants. And some of them are the vegetable that we eat. Lettuce and tomato. has been found to contain traces of CTCs, chlortetracycline, tylosin, and sulfamethoxine. Carrots, lettuce, and radish was found to contain gentamicin and streptomycin. One paper uh, reported the presence of tetracycline and amoxicillin in lettuce and radish. Another paper that works on radish shows the presence of chlortetracycline and rofloxacin and sulfatiazole. And this pak choy cabbage, this is the petchai that we have, they found to contain sulfadiazine, sulfamethoxine, and sulfamethoxazole. Okay, and I have uh, personal experience on this study when I use these pinto beans. And what I have with the pinto beans, we try to look at how tetracycline and chlortetracycline affect the growth of the pinto beans. Pinto beans Phaseolus vulgaris, like mungo. Okay, and we found out the mechanism why it has this phytotoxic effect. So we're able to uh, publish the results that we have. Okay, development of rapid pyrolytic assay to determine changes in relative levels of intracellular calcium in leads following tetracycline uptake by pinto bean plants. So what happened that we have there? So we let pinto bean grow in materials that contains antibiotics and one that contains just water or the other nutrients that it needs. The only difference, one has uh, tetracycline in it. And what we found out in terms of the growth, so this is the normal one, and this is the one that was treated with tetracycline. And we found out the mechanism. The tetracycline bind with the calcium that it has there in the plant. So the reduction of the calcium affect the growth of the pinto bean. Okay. Now, if we're going to look at the impacts of these antibiotics, I think the main thing that we need to worry about is the resistance, the so-called microbial resistance. So environmental levels have been found to enhance the ability of microorganisms to develop antibiotic resistance. And then there's evidence of antibiotic resistant microorganisms has been found in surface water, groundwater, and drinking water. Okay. So this is the most important thing that we have to look at. This is the effect. Okay, so what I'm trying to do here in this talk is what's the possible way for us to remove the antibiotics? Removal is the antibiotics is the only solution here, because if there is no antibiotics, then there's no chance for the development of the antibiotic resistant genes. Okay, so if we're going to look at the how antibiotic resistance happen from the farm to the table. So I have this, I don't know, photo information or some odds that they put out. So the way how resistance, okay, how animals can carry harmful bacteria in their intestines. So when antibiotics are given to the animals, so what happened? Not all organisms are killed. Those that survive, that's the one that is the resistant bacteria. Okay. And what happened? So if it's present there, okay, the animal products that you were there could carry that resistant bacteria. Or if you're going to look at the waste and then it's being introduced to the environment, some of the produce can be contaminated, okay, through the soil or water that was contaminated by the waste coming from the animal. And then you can also spread this resistant bacteria by prepared food through contaminated surface. Okay, so the environment when animals poop, and then after the spread, you will be exposed. So the contaminated food that you have, so people can get sick with the resistant infection. And then what will be the impact on that? There could be some resistant infection, and they found out it could cause you mild illness. To some, it can cause severe illness, which lead to possible death. About 1 in 5 resistant infections are caused by germs from foods and animals. So the problem that we have right now is these superbugs, and farm is one of the contributor of these superbugs. So if we put them in a growth promoter application of antibiotic, or we use antibiotics as a growth promoter, okay, the low doses of antibiotics that we give them, it will allow the growth of this microorganism, okay? And in doing that, you're developing an antibiotic resistance in the gut of the animals. So what happened? The bacteria from the animal gut get into the meat during the slaughtering and processing, or its waste would contaminate, okay, the farms that you have. And other organisms like flies and farmers carry it away. And what happened? You're going to contaminate the environment that you have there. So either contaminated meat or contaminated environment that contaminate other food products that was eaten or ate by the people, okay, that could give you sickness. So this is the main problem on antibiotic resistance. And this is not new, okay? They found out that bacteria has gained resistance to some drug therapy. And these are just some. So penicillin resistant, okay? These are some microorganisms, the pneumococci for pneumonia and meningitis, Legionella, the Legionnaires disease, Borrelia burgdorferi, the Lyme disease, the Salmonella, then the Staphylococci, and then the E. coli. Even us Filipino, we have this, the one that caused the tuberculosis. Okay, so there are a lot of problems that we have in this so-called super bug. And there's one book, they said winter is coming. So this is the winter that they have the development of this super bug. So why are we need to be concerned on these so-called super bugs? Okay, so if we're going to look at what happened, 80% of the gonorrheal infections are now resistant to antibiotics. 440,000 new cases of drug-resistant tuberculosis annually. So in the 1990s, what they say is the community associate outbreaks among the high-risk population. MRSA, okay? MRSA is very difficult to treat, and it persevered as a hospital-derived infection. In 2005, MRSA caused more diseases in the US than AIDS. Okay, so infections accounts for majority of skin and soft tissue infections treated in the emergency rooms in the US. And then we have here the enteric pathogens such as Salmonella and the enterohemogenic E. coli. They cause disease and treatment failure. So here in the US, we always have news where products are recalled because of E. coli or Salmonella contamination. So just like this one, 15,000 pounds of raw ground beef and pork products produced by an Oregon company was recalled because of the presence of the Salmonella E. coli. Okay, so if we're going to look at the problem that we have here, they said in the near future, a problem of a strep throat can cause you a scraped knee, and that could kill you. A simple hip replacement would result in 1 out of 6 deaths. Why? Because of the associated resistance. So the cost to treat drug resistant disease or drug resistant cases is estimated to be at least going to double in the succeeding years. Okay, so if we're going to look at how is this resistant form, So they said there are three processes that are responsible for this. Okay. So the first one that we have here is the conjugation. So we have conjugation when there is a direct cell-to-cell contact between two bacteria which need not be closely related, and transfer of small pieces of DNA called plasmids. Okay. And this is thought to be the main mechanism of the horizontal gene transfer. So this is the conjugation, just the transfer of plasmid. Okay, so that's just one way of developing resistance. The other one is transformation. So this is the one that you have here. So this is what? This is a process where parts of the DNA are taken up by the bacteria from the external environment. And then this DNA is normally present in the external environment due to the death and lysis of another bacterium. So there is a transfer, okay, of pre-DNA that you have here. Now the last that we have here is transduction. So this will occur when bacteria-specific viruses like bacteriophages, as is shown here, transfer DNA between two closely related bacteria. So this is how you develop resistance. So there are already some multi-drug resistance organisms, the Klebsiella pneumoniae for pneumonia, Acinetobacter baumannii, and then the Pseudomonas aeruginosa. And the relative few pathogens in nature, but we could say just treat that all antibiotic resistance is transferable because of these processes that we discussed. So imagine, okay, the need for us to develop ways to remove antibiotics, especially in bodies of water. And that will be the main topic that we have. I just try to introduce to you how antibiotics are present or introduced in the environment. And the next thing that we're going to do in my second part of a video is what are ways wherein we can at least remove antibiotic residues from bodies of water, especially the wastewater treatment plant that we have. Okay.